Okay, I'm going to go ahead and get started, everyone. I think I've met basically everyone here so far, but if I haven't met you already, my name is Mark Howen. I'm the Interim Scientific Director with GRIA and GRIN. And we have a wonderful panel or list of speakers here today to talk to you guys about similarities and surprises among our different genes that we look at in our foundation, represent our foundation. Welcome in, everybody. We're just getting started. So to kick off today, I'm actually going to start by giving you guys some insights from our recent GRIA census. It's just been such an honor to be here and to work with you guys recently and look at this data and now have the opportunity to interface with all of you. So thank you. So GRIA launched our census in August of 2024. With the goal of collecting data from families that can be used to get a better understanding of your experiences and priorities. This is really extremely valuable data, and this will be so essential for bringing us closer to treatments for GRIA disorders. And so currently the census is available in all of the languages shown here at the bottom. But we are also later this year adding Arabic and Hindi translations, so be on the lookout for that. We have had great participation in the GRIA census so far. We have about 441 individuals with GRIA reported, and so thank you guys for filling out the census. And for those who have not done so, you're welcome to. We are still accepting applications or participants, so be sure to fill it out and get counted if you haven't already. To tell you a little bit about the data set that I'm going to present to you today. So as I mentioned, there were 441 respondents. We did, for now, remove 114 variants of unknown significance from that. However, we will continue to update our analyses as we continue to characterize these VUSs. So from there, we performed these analyses on 327 responses. And I do want to make a note that for the data that I'm showing, if there were fewer than 3 responses for the individual gene, that will not be shown in the individual gene layout. And so that's specifically for GRIA 4, but that is included in the whole cohort analysis. And I also want to make the note that this data that I'm showing is representative of the people who have filled out the census, but may not be reflective of GRIA disorders as a whole, especially for individual genes where there's low sample sizes. Okay, so first I want to show you some of our census demographics. As you can see here, there are different numbers of individuals with different GRIA disorders. And so GRIN2B is up here at the top with 116, and then we have smaller numbers here, but those are continuing to grow in some of our things. We also have a distribution of sex, which is basically right down the middle. We have 49% male and 51% female. And we have a range of ages represented in our census, with the majority of participants being under the age of 10. Here I'm showing you guys a map of the locations of individuals who filled out this census. And so we can see we have a ton in North America, but we also had great participation from Europe, Asia, Australia, and South America. And now the number one thing that we wanted to get out of the census is really understanding your experiences and your priorities. So to start out, I'm going to show you a lot of graphs today, but I will hopefully take you through them in a way that's comprehensive. And if you have any questions, please feel free to ask me. This graph here is showing you the different symptom domains that we asked about along the y-axis here. And then the percent of participants in each of these different groups. So this is for every individual who filled out the GRIA census that we included in the analysis. In the dark purple, we have experienced in the last 30 days. So that's people who are currently experiencing this symptom. We also have in the orange experienced ever. So if you've ever experienced it, perhaps it's controlled by medication or things have changed. And then that's an orange, and then in the light blue is experienced never, and gray is unknown. And so from these data, we can see that it's really easy to see that fine motor delay, speech delay, and developmental delay are experienced by over 90% of our patient populations. So these are the most common, whereas things like maybe breathing challenges, hearing impairments, and dysautonomia are experienced in more like 30% or a third of our population. We can also break these symptoms down and look at them by gene. And so here I'm just showing you the top 3 most commonly experienced symptoms for each gene. And you can see in red those 3 most common experiences for our overall group, the speech delay, developmental delay, and fine motor delay. And so those come up in our top 3 for all of our genes with the exception of GRIN2D, but that's still very, very highly reported in GRIN2D. And so to look at that a little bit more as a whole, and I do apologize for the size of some of these things here. It's a lot of wonderful data on one slide. But this is breaking it down. So now we have the different genes across the x-axis. And again, we have our domains over here. And the number that you're seeing in the middle is the percentage that has experienced it. So this is including current and past. Overall, you can see very similar trends for some of these. We have really high numbers and percentages of individuals having experienced at some point some of these motor delays and speech delays. But we can also see differences from this type of data. And we can see here, for example, that with hearing impairment, individuals with GRIA 1 are experiencing hearing impairment much more often than some of the other genes, sometimes 3 to 4 times as many for the percentage. But I want to break down a few of these further. So for example, epilepsy rates. We have about 50-50 of individuals who've experienced ever or compared to individuals who maybe do not have seizures or are unknown for if they have seizures. We do have variability also in the types of seizures that present. So the highest here is tonic seizures and absence seizures, but we do have quite a range. However, if you look at that by group and by gene, you actually see a lot more of a clear and interesting picture. So it's not that 50% of all individuals for all genes that are experiencing seizures, but you might have some groups, for example, GRIN2D here, that have closer to 90% seizure rates, whereas with GRIC2, 17% of individuals who fill out the census have experienced that. And while epilepsy can be quite different among the genes, muscular issues are more consistent. And so 87% of our cohort have experienced muscular issues, either currently or in the past. And this is overwhelmingly, most commonly hypotonia, although we do also see hypertonia as well as others. And this also holds true for across the different genes. And so here again, the genes are listed on the y-axis, and then we have the different types here. And you can see in the darker purple, which is a higher percentage, that there's a lot more hypertonia than hypotonia than hypertonia. I'm sorry about that. And you can also see more consistent rates of epilepsy across the different gene groups. So this is much more similar in terms of percentages than what we just saw with epilepsy. Now I'm going to briefly talk about co-occurring conditions, which are also known as comorbidities. And so over 60% of our participants have been diagnosed with global developmental delay in addition to GRIA disorder. But individuals may also have a diagnosis of things like autism, ADHD, cerebral palsy, and Lennox-Gastaut. And you can see those percentages for our overall group here. And I'm also showing you now by gene the top 3 most common comorbidities, so global developmental delay, autism, and ADHD. And so for all of our genes, we have really high percentages of global developmental delay, and it's higher than the other comorbidities, with the exception of GRIA 1, in which autism is actually a higher percentage diagnosed. So in line with this high percentage of individuals that are experiencing global delay, what I'm showing here is reports from individuals who filled out the census about how the person with GRIA compares to their peers in the areas of intellectual and cognitive abilities, speech abilities, and fine motor skills. And so in the red here, we're seeing severely behind their peers, in the orange, moderately, yellow, just a little bit, and then green is the same or better than peers. And you can see across these 3 domains that the majority of individuals across all gene groups have been reported to be severely or moderately behind their peers in these areas. But despite these delays, many individuals do achieve developmental milestones, especially in older children. And so these data here are the percentage of individuals reported to have achieved the milestone at the time that the census was completed in a singular data point. And so these are now broken down into different age groups rather than different genes. So across the bottom, we have age 0, 2, 3, 5, up to 18 plus. And then we have the different milestones that we probed, with the darker color and the darker blue being the higher percentages. And so while things like sitting up independently and supporting weight have really high achievement rates, especially in older individuals, things like toileting and communication have lower milestone achievement rates, even in older individuals. And so this was singular, but I want to make sure that that's clear. So if you see data points like this, where you see a higher percentage of individuals in this lower age group compared to this older age group, that doesn't indicate a regression, because we are probing at a single time. When you're completing the census, we're not doing longitudinal data. But I do want to make a pitch for the importance of longitudinal data, so we can learn things about things like regression. Okay, and lastly, I'm going to break down these milestones by gene. And so we do see differences in the individual genes in terms of achievement rates, as well as in individual skills. So here I'm showing you the different median milestone achievement by gene as a percentage. And then here I'm showing you again the milestones we probed, but now across the bottom are the specific genes. And so you can see the achievement rate for the individual instead of the whole population. And for timing's sake, I'm sorry that I had to speed through all of that. But just quickly to conclude, what we're seeing is that developmental speech and fine motor delay are the most commonly experienced symptom domains in the whole GRIA community and across gene group. We're seeing that rates of epilepsy and types of seizures do differ significantly between individual genes, whereas things like muscular issues are more consistent, and hypotonia is the most predominant muscular issue for all groups. Individuals with GRIA disorders do often have one or more co-occurring diagnoses, with global developmental delay occurring in 60% of census participants. And individuals with GRIA disorders do reach many milestones, although achievement or rate of individual skills can be really variable and differs by gene. So with that, I just want to thank everybody so much who has participated. This is not possible without what you do. Thank you also to the GRIA and GRIN team, especially those who have hunted down people to do the census and fill it out. Thank you to Chan Zuckerberg Initiative for their help with some of the data analysis into Combined Brain. Thanks. And now we're going to go ahead and move into our second speaker. So we're going to have Dr. Kristen Parkham to talk about her work as well. Thank you.
Hi, thanks so much. This was the title that I was kind of told for the presentation. So I kind of turned it into a little bit of a battle maybe between GRIA and GRIN, and you can see, who's going to take over the world. Unfortunately, when I went to prepare the slides, I realized I did not have as much about GRIA disorders. So we'll see how far we get. So next slide. What? Oh, right, I can use the thingy. That's right, sorry. So first, I think a little bit of clarification about what GRIA and GRIN do, because I think we've had a lot of focus on GRIN genes when this all started, but I think these have been added more recently, so I just wanted to go over what they do. They mediate a fast component of excitatory, so kind of the gas of the brain versus the brakes of the brain, transmission of signals between neurons, and most of the neurons within our body are excitatory, and the synapses are excitatory. So this is a really important area for communication. These genes combine with a number of other these proteins that are made by the GRIA genes combine with a lot of other proteins and get embedded into this network of scaffolding in the synapse, and they also can signal other proteins down the line. They are involved in synaptic plasticity, which is a process that is how we learn, meaning that synapses that fire together kind of become stronger, and those that don't may become weaker, and that's the basis for learning and memory. Most of these genes are not very tolerant to variation. So if we think about our genetics and our differences, we may have about 3 million differences in how our genes are spelled between any few given people. But those differences are not spread out evenly along the genetic material. They're concentrated in places where they are tolerated and they don't cause any problems. And different genes in our body are tolerant to different degrees of variation. And GRIA genes are very intolerant to variation. And there are several kind of what are called hotspots, meaning places where variants that cause problems cluster. And that's not true for all genetic diseases. It's true for some, but not all of them. And that's, I think, a difference with GRIA that is not always seen in the GRIN variations. And I think there are fewer variants that are known, only about 100, and they are more challenging to categorize as far as how they affect how the gene and the protein will work. And I think that's what Dr. Coombs and Dr. Bayat's lab are working on, is how do we get that information for this newer class of genes. So to provide a little bit of background and why we're still at that point, I think, is because many of these diagnoses were only described, a few years ago. So the first paper coming out for GRIA was in 2019, when it was seen in a microdeletion, that meaning a larger piece of genetic material was missing, and GRIA2 was one of the genes within that section of genetic material. And they thought, wow, I guess that's kind of a good candidate for maybe what's causing the problem, because we know that NMDA receptors are involved in neurodevelopmental disorders. Maybe AMPA receptors are too, and maybe this is what's causing that child's symptoms. So then they kind of went on and kind of did some more work with this and developed some mice. And then they also were able to subsequently publish a larger case series of patients. And that's kind of how most of these genes and our knowledge about them get started. We have one patient, we think, oh, I wonder what's going on there. And then, people kind of come together and bring us more patients, and then we work out the mechanism, and then we can start getting work on what's going to help with treatment. So I think what we learned from that was that, these patients had very similar problems to what we've seen before, and that also it's going to be challenging to sort out the functional significance of some of these variants, because even variants that were right next to each other had very, very different properties in how the proteins ended up working. So I'm going to present some data from the US registry, and unfortunately, again, we only have 9 patients from the registry, so that's not very much data. But you can see that GRIA 2 and 3 are most represented, with several patients in each of those, and then much less data for 1 and 4. 2 of the variants were listed as being inherited, and I think we have seen that, especially in GRIN 2A. And epilepsy was present in about a third. Again, we saw some spread and what the incidence of epilepsy is between these various genes. So there were about a third of patients who had a diagnosis of epilepsy, not necessarily that they were having active seizures, but that they have had a diagnosis of epilepsy. There were a couple patients who were being treated with Fycompa, which I think is interesting because the mechanism of action of that medication is to damp down AMPA receptors, and so I think a kind of a targeted therapy, and I think Dr. Coombs and talked about that today a little bit. We have EEG data from a few patients, and only one of them had an abnormal EEG, although there were a lot of missing data points there, which is why I put that little asterisk. We kind of look for several different EEG patterns, one of them being this spike wave activation in sleep, otherwise known as ESES or CSWS, and that was not seen in any of these EEGs. We had some patients who had infantile spasms in the previous series, and hypsarrhythmia is the EEG pattern that goes along with that, and we didn't see that either. And then we also, in the developmental and epileptic encephalopathies, can see slow spike and wave. That's a particular sign of LGS or Lennox-Gastaut syndrome, and that was not present either. So these are our total numbers as of the end of 2024 for both our registry in the United States and in Leipzig with Dr. Lemke. And you can see that the GRIN genes are certainly much more prevalent out of our total of 1,000, with only about 71 GRIA genes, most of them being in the EU or the areas that registry serves. And so I think Dr. Bayat is leading the functional testing effort, and so I think we're going to hopefully try to coordinate and get him all of that data and make sure that we're communicating well. This was a slide that the Leipzig team sent me about GRIN, so moving on to that and talking about what we've seen in our data from that. And so these are the major categories where we had a significant number of patients. So you can see the, oopsies, I meant to do the numbers there, 187, 248. This is quite low. I think we have a majority of our patients with GRIN 2B and then very few GRIN 2D individuals. And again, these are patients with pathogenic and likely pathogenic variants. So that excludes those that are uncertain. And so you can see again, developmental delay, a very common presentation, maybe less so in GRIN 2A, which I think is something that we've seen several times, that spectrum of learning and cognition can be quite a bit broader in GRIN 2A, and that's one of the ones that can be inherited. And a lot of abnormal MRI in GRIN 1, GRIN 2D, GRIN 2B, less so. They didn't put a data point there because I think it's quite low in GRIN 2A. And then the incidence of seizures ranging from 100% in the GRIN 2D patients, although that's only 15 people, to the lowest being, in the 40s for GRIN 2B. So this was the data that I pulled from our GRIN patients. We have about 97 total. Our most common set of patients is GRIN 2B, and I think that's probably true for most genetic testing kind of results. We just enrolled our first GRIN 3B patient, which is very exciting. We mostly have missense variants, which I think is the ones that are most challenging to interpret, also the ones that have the greatest potential for being either loss or gain of function in their in how they affect the protein, and most of them being de novo again, except for GRIN 2A and a couple of GRIA variants. Movement disorders were present in a lot of the patients that I queried, the most common being stereotypies, so things like rocking or hand flapping or kind of excess excited movement, so to speak. Autism spectrum disorder present in a relatively low number, but you can start to see that we have a lot of missing data points here. So probably for each of these fields, there were about 10 or more fields that weren't completed. So the numbers are not the same as the total number, but you can see again that intellectual disability is quite common, and grading that intellectual disability, most of them had severe, or half of them had severe intellectual disability. Cortical visual impairment, which we've talked about a couple times at this seminar, was present in maybe a little under a third, and that is a very specific type of visual impairment. We asked questions about other types of visual impairment, and so we got information about ocular misalignment, which again can lead to vision problems if the eyes are not receiving the same data from the world. They tend to ignore one of them, and then vision can be compromised. Nystagmus or jiggly wiggly eyes. A lot of kids needed glasses, and then some kids had this very specific type of eye movement called an oculogyric crisis, which is sometimes classified as a movement disorder, but sometimes classified as a visual issue. And then we had storms present in, not as many people as I think we had anticipated. Next, we look at some behavioral issues, and I think the most common was probably about half, most common being irritability, and then kind of less frequent as you get down the line. And about 10 patients had symptoms that were severe enough that they had at some point in time used some medication, and that was most commonly for aggression. Obviously, if we think about what's going to be most damaging and disturbing to the child and the rest of the family. About 19 patients were using a precision therapy, so something targeted towards how the NMDA receptor works. And so for GRIN, that was something like memantine or L-serine or magnesium, something along those lines. And I thought that was great to hear that people were giving those things a try. The MRIs were largely normal, but again, that's a lot of missing data points. But there were a malformation of development in about 8 patients, and we ask about more than one MRI, and so sometimes the first MRI was reported as normal, and subsequently a malformation would be identified, and I think that's an important clinical point because kids who get MRIs at very young ages, it's very difficult to see some of these malformations, so especially under the age of 3, so if an MRI is done at that age and there's still some concern about seizures, it may be worthwhile to talk to a neurologist about getting another scan. As I said, 65 out of 81 were treated for seizures. When I look at the medications that were being used, nothing really stood out as being the most effective. Most of the patients had convulsive seizures. That was the most common seizure type that was reported. And then only 3 patients, which I put a question mark because I think that's probably low and maybe reflective of not asking the right question, but 3 patients were reported with this spike wave activation in sleep, which is most common in GRIN 2A, although I did just see it in a GRIA patient in our clinic a little while ago. So this is some information about the registry, and we have the contact up here. So if you are residing in Europe, Asia, or Africa, or other places that are not on this list, then you would go to the Leipzig registry with Drs. Lemke and Cray. And then if you are in the Americas, North or South America, or Australia, you would be in the our Denver registry, and then there's also a registry through Simons that we try to coordinate our data with, and that's the website for that. And then to find your information about your variant, this is the new portal website that Dennis manages. So that was it. Thanks.
And just to finish up these last session here, we're going to end with Katina Kalakos telling us about Simon Searchlight.
All right. Thank you so much for having me. I'm happy to present here for Simon Searchlight and what we've learned so far in the GRIA registry. So Simon Searchlight is an international research program, and our mission is to accelerate science and improve lives for people with rare genetic neurodevelopmental disorders by collecting high-quality, standardized natural history data and building strong partnerships between researchers and families. We are fully funded by the Simons Foundation Autism Research Initiative, or SAFARI, which gives us the opportunity to do what we do here. So first, we collect detailed medical and behavioral histories along with optional blood samples. We then synthesize that information provided and share those results back to you as the families. We also share de-identified data and samples with qualified researchers because we want as many professional eyes on the data to access, analyze, and then publish in the literature, really driving those advances forward in science and medicine. We also want to connect researchers that are outside of Searchlight with participants from around the world to pursue other research endeavors. And then all of this really is to promote better understanding of these genetic variants. And so this is our long table of different online measures that we ask families to complete. And I want to lead by saying thank you so much for the time that you spend. We know it's a lot, especially when we ask you to give us updates every year. But it's really important to have those annual updates because then we can see what GRIA is looking like at younger and older ages. So right at the top, we do ask for a genetic laboratory report. So our genetic counseling team can make sure you're in the right place, we have your variant correct, and then that unlocks the rest of the participation in the study, ranging from surveys online such as medical history data, seizures, sleep, development, behavior, communication, quality of life, and more. As I mentioned, we also try to connect other researchers with families to further pursue more research opportunities. And so this is formally called the Research Match Program. And so if there are families that are eligible to participate in someone else's study and that researcher lets us know about it, we can try to work with them to get in contact with families that might be interested. And so over 3,000 Searchlight families have been invited to participate to date. And as I mentioned, ultimately, all of these data are then de-identified and available to researchers with qualified access through our data distribution platform. The goal here being to analyze and publish on it so more people better understand GRIA. And so with Simon Searchlight data across our full cohort, we've had over 130 papers that have used Simon Searchlight data in some way. I went through the list to try to identify all that specifically were interested in GRIA or used GRIA in combination with other conditions, and I found at least 9, but I will say that might be an underestimate because sometimes it's hard to query exactly where all the data are living. But at least 9 have specifically looked at GRIA data, and then we also have 3 research match studies that have recruited from different GRIA communities. For the families to know that your data is being used effectively, and also for researchers who are interested, we did just put out a new refreshed data set on our data distribution platform called Safari Base just last month. So our updated numbers are 231 GRIA participants. This is across VUS, likely path, and path variants. And then more globally across the cohort, closer to 5,700 people with genetic variants studied in Simon Searchlight. So if you're interested, please let me know. I can help sort that out if you don't know how to access. And then there's also a new grant opportunity coming out of Safari, which is our funding agency. And essentially, the details are there. I recommend just going to the website for more information for the sake of time. But there is opportunity for analysis of pre-existing data sets, including Simon Searchlight. And so that'll be due later in September. Okay, so now I'll dig into the data on what we've collected so far in the GRIA registry. So this is a bit of a busy slide, although it is one of my favorites because I get to see the numbers change every year. So from step 1 through 6 are the different steps of participation in Searchlight, registering, consenting, providing lab report, giving data, et cetera. And then each row is divided by the different GRIAs that we have in Searchlight. So we have GRIA 1, 2, and 3, GRIC 2, and then GRIN 1, 2A, 2B, and 2D. I'll note 2B is our largest community in GRIA, followed by GRIN 2A, GRIN 1, GRIA 3, and then we're growing our numbers in some of the other GRIAs, 2D and GRIC. So this and the next few slides, I won't spend too much time on this for the sake of time, but I did want to highlight both for the families to understand and for the researchers in the room to see the different spectrum of variants that we have represented in Searchlight, and I can make some of these slides available as well for follow-up. All of the slides will look something like this, where in the text, we're seeing the different variant information and looking across the protein product where that genetic variant would be impacting. And so we did also try to cross-reference with the GRIN portal where possible what functional information was available for particular missense variants, and that's been color-coded. We try to keep updating this every year to better understand what variants we have represented. So here we have 6 participants with likely path variants in GRIA 2. And then I'm going to just speed through these, sorry about that. We have 7 participants with GRIA 3 likely path. Again, we're growing these numbers. Currently, we have 23 participants with GRIN 1 path or likely path variants. This is 5 more than our last meeting, so this group is growing as well. We're not showcasing variants of unknown significance, although all of that data, again, is made available to researchers. We have 22 participants with GRIN 2A likely path variants, and this is 8 more than our last meeting where we presented. And then again, our biggest group here in GRIN 2B with 139 participants with likely path variants, 16 more from the last meeting where And with the color coding here, we can see we skew more towards loss of function for those that are known functional effect, a few likely possible gain of function, and then many still to be determined. And then finally, with our 2D group, a smaller group here, we have 4 participants. I mentioned we are, by virtue of being online and GDPR compliant, we do have representation across the globe. We are working on building out our battery of surveys to be maximally available in different languages. So we're currently offering at least something in English, German, Spanish, French, Italian, Dutch, and Portuguese, but we are trying to increase the number of surveys that we're having translated. I mentioned as well that we do have an optional blood sample component to Searchlight. I won't dig into this too much, but we have had many families from the GRIN2B community who have donated blood, as well as families from GRIN2A, GRIN1, and then one from GRIA 3 and one from 2D. The blood samples are really helpful because researchers can use them to better understand the particular genetic variant. And in some cases, those blood samples can also be used to create induced pluripotent stem cells, or iPSCs. Selection for this is based on scientific and clinical priority, but it essentially allows the researchers to model more closely what those cells look like to better understand the biology. And so from the very generous contributions of GRIN2B families, as well as the collaboration that the Simons Foundation has with the Nancy Laurie Marks Family Foundation and the New York Stem Cell Foundation, there have been 12 variants of GRIN2B that were identified and selected for reprogramming into stem cells across 13 families, since there were 2 carriers with one common variant, with the one same variant, sorry. And so most of those were successfully reprogrammed and available to researchers. We are trying again for a couple of them. And so far, 15 research groups have already requested access to these. And if we dig into the data a bit, I'm going to start mostly with GRIN2B, and then at the end, I will have some preliminary insights on our growing numbers in GRIN1 and GRIN2A. But I will caveat again, much more preliminary than some of the other researchers working in those communities. So not exactly an even comparison. So for GRIN2B, we have 109 individuals who've contributed medical history data, about evenly split by sex, ranging in age from 1 year through to 38 years, with an average age of 9 years. And most of the rest of the slides are going to look something like this. So just to quickly orient you along that vertical line or y-axis, we're seeing specific kinds of diagnoses that we ask about in our survey, saying, has your GRIN2B loved one been diagnosed with this at some point in life? And so, for example, if we look at that middle yellow bar, the longer the bar, the more people are reporting having that diagnosis. So we're seeing nearly everyone in Searchlight with GRIN2B is reporting intellectual disability or developmental delay, seeing similarly high numbers in language delay and impairment, seeing some folks with autism and ADHD, smaller subsets, about 20 to 30%. And then if we're looking head to toe at the different body systems affected medically, this chart is organized where the body systems at the top are overall less affected based on reporting from GRIN2B families, and those that are at the bottom are more affected. So if we kind of zoom in to the bottom here, we're seeing mostly neurological, GI issues, vision, endocrine, orthopedic concerns, and then less so in some of those domains further at the top. And then in the brackets, we're seeing just for those that are maybe less represented, what are we seeing most commonly? So, with joints, we're seeing mostly hypermobile joints. We're seeing instances of chronic fatigue. For orthopedic, we're seeing higher reports of scoliosis. And then in our GI, mostly GERD and constipation. And so if we dig in a little bit to what parents are reporting for neuro conditions, we're seeing very high rates of hypotonia or low muscle tone. We're seeing coordination problems. We're seeing movement disorders in GRIN2B, and I've just highlighted there some of the specific ones that have been cited by parents. And then an additional, several other different neurologic conditions that are affecting smaller samples of people with GRIN2B. We do also collect data on seizures. So the left panel is showing from our medical history survey the different subtypes of seizures being reported by families. And then to the right in the box, from a different, deeper dive survey into seizure history, we're getting some metrics about things like when are seizures starting, how many people are taking medications and are they continuing to take medications. And so we're seeing seizure onset based on caregiver report ranging from 1 month through 12 years of age, averaging out about 2 to 3 years. At least 2/3 are taking medications, likely more because sometimes families won't remember the meds or maybe they don't have their report on them and so they may skip those questions. So I'm going to guess that that number is actually probably higher. And then in terms of reporting seizure control, about 1/3 of families are reporting feeling like they are having some control over the seizures with a similar age range to age of onset, 9 months to 12 years. Vision issues also comes through commonly, as has already been shared in the GRIN2B data in Searchlight. And so here are a number of different vision-related concerns that we ask about in the GRIN2B community. And then we also ask about medication use, specifically what kinds of medications are you using for what and how effective is that for treating what you're hoping to treat with the medication. So this doesn't have all of that detail in there, but we're seeing commonly reported meds used for helping with GI issues, with seizures, with sleep. We did ask specifically about seizures, if there was a single medication that seemed to be most effective, and not surprisingly necessarily, there wasn't a single medication that stood out across 2B, but we do have families reporting that they're finding what's working for them and other families that are still looking to see what will work for them. And with my last few minutes, I will show some preliminary insights on what we've learned from Simon Searchlight data in the GRIN1 and GRIN2A communities. Again, highlighting preliminary since our numbers are still smaller in these communities. And so in GRIN1, we now have 14 individuals with pathogenic or likely pathogenic variants, and they are about evenly split by sex, with average age being about 10 years of age. Similar setup on the graphs here as previously with GRIN2B, also seeing higher levels of reporting of intellectual disability and developmental delay. We are seeing language delay and impairment, but to a lesser degree in this earlier sample than compared to 2B, for example. And similarly, we're also seeing lower reporting of autism and ADHD, but not zero. And I think if I understood some of the other data presented, I think there might be some alignment there. So that is good to see alignment between the data sets. In terms of head-to-toe medical issues for GRIN1, we're seeing a lot of common body systems affected, again, neurological, some endocrine, some GI, and vision. We are seeing seizures as well in GRIN1 with those subtypes listed there. And similar common conditions through those body systems coming through in GRIN1 as we saw in 2B, like hypermobile joints, GERD, constipation, failure to thrive. And if we dig a little bit into some of the neurological conditions we ask about, similar to what we saw in the 2B group, we're seeing low muscle tone or hypotonia, similarly seeing some coordination problems, and then also seeing percentage-wise, I think, more in the movement disorder. But again, this is a small group, so it might be a sampling effect. We would need to kind of see that out more as more families join. And then finally, I'll end with a couple of slides on GRIN2A, an even smaller group, our 11 GRIN2A families, thank you, with likely path and pathogenic variants, evenly split by sex roughly. Again, 8 months through 23 years of age with an average age of 9 years old. Similarly, again, seeing language delay and impairment coming through as commonly reported, intellectual disability and developmental delay. And then we're seeing a higher amount for autism and ADHD than we saw in GRIN1, closer to the estimates that we were seeing in 2B. High level, a lot of the same conditions, body systems affected that we saw in the previous groups, neuro, gastro, we're seeing seizures as well. Between the three, this is our group that is reporting the highest percentage of seizures. And again, to better understand the data that I've been hearing throughout the conference, that might continue to pan out as we get more families enrolling, but we'd want to see more families to see if that persists in our data. And then again, in terms of neuro, we're seeing low muscle tone coming through most commonly, movement disorders, coordination problems. And so to close, I just summarized a bit about looking across the 3 GRIN communities that I show data for today. So at the top here, across all 3, we are seeing reports of IED and developmental delay, language delay, GERD and constipation as key GI issues, varied vision conditions, so not exactly the same across, but vision system affected nonetheless. Most of the groups are reporting high levels of low muscle tone and other varied neurological conditions, and we are seeing seizures throughout, but the degree of seizures is variable depending on which community we're looking at. And then if we look to see what might be not necessarily common across all 3, but maybe in a couple versus the other, we did see things like failure to thrive and scoliosis being reported mostly in the GRIN 1 and GRIN 2B and less so in our GRIN 2A community. And then in terms of the right-hand side, we were seeing higher reports of autism and ADHD in GRIN 2B and GRIN 2A and less so in GRIN 1. But again, just a caveat, we really do need more data across all of the GRIA communities for a more complete cross-comparison. And additionally, I'll note, and this has come up a lot throughout the conference, and it's very important, that the function of the variant really matters biologically and clinically. And here we're pooling together everyone, for example, with a GRIN 2B variant. But it'll be important for us to organize our clinical data by gain of function or loss of function as we learn more about that with all the great work that other folks in the room are doing. And so with that, I'd like to thank you so much for listening to me. Thank you to CureGRIN and GRICon for having me come and speak. And all of the families for participating. If you're interested, you can register at simonsearchlight.org. And for the researchers in the room, if you'd like to request data and biospecimens, you can head to base.safari.org or email me, because I love talking to everybody. So thank you very much.
But we are a little low on time, so we're going to keep it to a quick 5 minutes. Yeah, you guys go up there and I'll pass the microphone and then I'll join you up there. And I'll have Bro to the microphone.
Hi, I think this might be a question best suited to Dr. Park, but if I'm wrong, others please chime in. I'm wondering to what extent you can comment on the variability in numbers of cases we have so far across the GRINs, the GRIAs, the GRIDs and the GRICs. To what extent is that a combination of sort of tolerance to variation, either increased or decreased, because there's 2 forms of sort of selection pressure happening there, and the sort of recency of the characterization of the disorder and, the classic challenges related to that?
I think it's probably a combination of both. I think we heard a lot in the GRIA presentations that because that gene is relatively new, there's a lot of them that have been classified as variants of uncertain significance. And so I suspect there are probably families sitting out there who have that diagnosis but just don't realize that they have that diagnosis because nobody really explained it well enough, and they have kind of put that report to the side and said, well, that must not have been it, when in fact it may be. So I think there's some of that. I think, but I do think that there's probably some incidence issues, that some of the NMDA receptor disorders are probably overall more frequent than the other receptors as far as how often they happen. I think it has yet to be fully seen because I think we don't have enough data for some of these newer diagnoses.
I think my question is for Dr. Park as well. We've been using magnesium for a while with a GRIN 2B child, but I'm not sure why it seems to help. Can you explain why you would recommend magnesium and what the mechanism is?
Yeah, so magnesium is, NMDA receptors kind of depend upon magnesium. It sits there, and in order to activate the receptor, you have to release that block. And so I think in that way, it can be something that's targeted, but I don't know if we know, a lot about how much has to be given, or, again, what types of functional changes in the receptor best respond to the use of that GRIN-specific treatment. So I don't know that I can say a whole lot about it as of yet, because I think it has less impetus to trials behind it than some of the other things that we've been using, like Fycompa or Memantine or even L-serine.
This was a question really raised by the last speaker, and I just wanted to follow up, but it's actually for everybody, which is in terms of looking at the function of variants, have any of you really tried to have larger data sets to group rather than by gene, by loss or gain of function, either within a gene or across different genes, to see if you could pull out any the maybe more subtle phenotypes that are clearly associated with a subset of patients?
Yeah, that's a great question. I think the more data we collect, the more powered that researchers would be able to do that. Currently, in terms of presentations we present primarily to families, we're usually coming to meetings with a specific gene in mind, but a lot of the researchers that we work with are looking more cross-genetically and can kind of parse those relationships more effectively. So we would love to encourage as many creative and interesting analyses to look across that as possible.
I would say I was going to call out Dennis there because I think that's some of his research, looking at groups looking at channels and how do channels function and where do variants in channels tend to cluster if they are going to be gain of function versus loss of function, because they have that same kind of split / spectrum as receptors do. And so I think we're trying to figure out what are the commonalities between different types of genes that cause epilepsy and neurodevelopmental phenotypes, and kind of put that all together. And I think maybe Dennis, you want to comment on some of your research in that area?
Yeah, because we scientists are idiots, right? So you can just go to the GRIN portal and do it yourself. So you actually can't sort by function and see what happens on the clinical side. The only unfortunate thing is that the clinical side is a little bit poor on the portal, and I hope that we soon have more data to combine, because this is you see this there's so much happening, and every information is important. So, but you can actually do this yourself. You just go to the GRIN portal or go to the research section and you can filter by function. And yeah, I fully agree. Yeah, yes. But people are doing this. So in general, and because at the end, all these channels they work similar. They look similar, and but the only thing is where they are expressed and when they're expressed, and this can have clinical implications. And the only thing what I wanted to also add to the prevalence, there's a paper from Rivera Lopez from us from 3 years ago, and you can see the frequency from each disorder. And this is a genetic approach to estimating the prevalence, and is completely independent of all the biases which one have to genius not being on a panel of behavioral specialists are not doing genetic testing as frequent as neurologists and so on. And I think the GRIA genes are very similar to the GRIN genes, just because they look so similar, and at the end, size matters in this case, and also the constraint. So but anyways, so but the estimated prevalence estimates are in that paper.
Yeah, we have not done this yet, but this is something that we're interested in doing, is looking at gain and loss of function for those that have been characterized. Of course, that's the huge caveat there. The other thing we want to do is look at it by sex differences, not just by gene. So we have that data. We haven't analyzed it yet.
But I would also say that I think it's important to not make things too simplistic, right? I mean, I think we heard earlier today from the SCN2A mom who spoke that, we had a lot of data and we said, oh, we figured it out, right? If your epilepsy starts before a year of age, then you clearly have this particular type of functional change. And if it starts afterwards, then you have this other. And that, obviously the body is more complicated than that. And so that turned out not to be entirely true. And so I think it's still going to be pretty complicated to sort out what each of these regions means for how it affects the protein. Again, as Dennis was saying, not only do we have the location of the variant, we have when is the gene expressed in the body, what is it involved in, there's just a lot of factors. Other questions?
Hi, I'm Lana Wong. I have just a general question. Earlier today, Keith went over the different, data repositories of where parents could be uploading data. And I know that we had functional analysis done at Emory, and it's in their database. And I just assume that these other databases or whatever, that they're somehow accessing that information. So am I correct there? Or should I have been including that functional analysis if I've uploaded information? And I'm not even sure like what kind of report I even got, because they didn't want to give me the report directly. They said, you got to meet with the physician who's got to explain it to you. So I don't even know what I have.
This is a common thing and a lot of complexities, and I think it stems from kind of work starting in a lot of different places at different times and us trying to kind of merge it together. So I think we're really working on that. But there are also a lot of complexities to research and clinical care between different institutions and different countries. And so we are, the EMRI data goes into the registry, which I talked about, which also communicates with Leipzig, which kind of takes the rest of the world. I think the GRIA Census is slightly separate and requires a different entry. We try to merge with Simons whenever we come up with reports. Our groups, query the database and make sure that we're not double reporting people, and they have slightly different data set than we do. But I think we're doing our best to align so that everything is together. But because of funding and regulations and so forth, it gets pretty complicated to just put everything in one database that everybody can get access to all the time, right? That would be ideal, but very hard to do.
Right, and then you raise another point about potential double counting and ensuring that you're not double counting.
Correct, yeah, that's really important too. I think that's easier in some of the EU countries because they have a national identifier and they can use that to identify patients, but because of the United States, no, we don't have that kind of system, and many studies are trying to use de-identified data, so it's quite complicated.
Right.
Great, thank you to everyone, really appreciate it. We're going to move into our next session.